Hi everyone, my name is Stephanie Whitson. This is a lab presentation for General Chemistry 2. In this experiment, we are taking gummy bears and we're going to be putting them in tap water, and then we're going to be also soaking them in a saturated salt solution. And then primarily, this is going to be an example for osmosis, which is the movement of liquid by diffusion from an area of high concentration to an area of lower concentration. With the tap water and then our salt solution that we have, we'll talk more about that. I'm going to get this experiment set up and I'm going to pull it down so you guys can see what I'm doing. So we've got our 2 cups, one for our tap water and then one for our salt water. And basically what I'm going to do, I'm going to put water in each of these. And then we are going to take our salt and put in this bad boy here. So we'll put salt in this one. Once salt is put in there, we're going to stir it up. And we're going to stir that until we are unable to dissolve any more of the salt in there. So once that's finished, we are going to take our gummy bears. Just going to grab 2 red ones out of here and just kind of show you the size. Pretty solid, not very, flimsy at all, but we're going to drop these in there. Both of these have sunk to the bottom just right off the bat. So we're going to leave these for 6 to 10 hours just to soak, and we're going to see what happens with these. So basically our tap water solution is going to be what we would like to call more of a hypotonic solution. And in a hypotonic solution, the concentration of the solute is greater than the concentration of the solute in the solution. So at that point, it's referred to hypotonic, and the prediction is that this gummy bear in the tap water solution is going to swell and increase in size. Our concentrated salt solution is what we would like to refer to as a hypertonic solution. And in this, if the concentration of the solute is less than the concentration of the solute in the solution, then it's considered hypertonic. And the prediction is that the gummy bear will shrink. So we're going to wait our 6 to 10 hours and do it just like they do on Food Network and switch them out. So now we have our 2 solutions, and these have been sitting, like I said, for 6 to 10 hours. We're also going to pull our control sample out of the bag. You always want to make sure you have a control when you're doing an experiment so you can observe the differences. So now we are going to pull out. So just a quick observation, the tap water, the gummy is still on the bottom. We'll pull it out of there. It looks a little flimsy. But let's get that out of there. Move that out of the way. And then in our concentrated salt solution, the gummy is actually floating. Just a quick observation of that. So I'll lay him down here. So as you can see in the hypotonic solution, definitely our gummy is much larger. he's a little bit more translucent, and he's kind of falling apart here. But yeah, you can see compared to our control, much larger. Definitely in a hypotonic solution, it tended to swell. So let's take our control over here. In our concentrated salt solution, honestly, they look about the same size. The gummy in the salt solution quite possibly could be a tiny bit larger. That could be because, perhaps I didn't put enough salt solution in there to pull, the water to the higher from the higher concentration to the lower concentration, so it didn't shrink it any. Or maybe it needs to go a little longer than 6 to 10 hours. That could be possible as well. But it just kind of gives you an idea, compared to just the regular tap water. The salt solution definitely is a huge difference, comparable by size. Yeah, and that's it. Fun experiment. Kind of shows you exactly how osmosis works, and if you apply this, to real life, we look at the living cell, the cell membrane that allows different molecules to come in and out. If you tend to have an offset of those, you could be like, for instance, if you're dehydrated, the water is going to move out of the cells, and the cells are going to shrink. So kind of an application for real life. Interesting project. Really enjoyed it, and I hope you guys did too. Thanks.